Welcome to this tutorial on animal and plant cells, and I guess my main objectives here are to develop with you an understanding of the subcellular structures, and secondly to encourage you to think about the different scales and sizes of animal and plant cells. So first things first, I'd like to introduce you, or really I guess reintroduce you, and I hope you've watched the eukaryotic and prokaryotic cell video already, but I'd like to introduce you to, first of all, an animal cell. And this animal cell has here, as we can see, has here what we're going to refer to as a cell membrane. A cell membrane. And remind yourself about the cell membrane. It is ultimately the feature that separates the cell from the external environment, from other cells, of course, and we would describe it as semi-permeable. Now, because it's semi-permeable, we can also say about the cell membrane is it controls the passage of gases and other materials. And we're talking about into and out of the cell. So it's the cell membrane which controls what goes into and out of that cell. Secondly, let's remind ourselves from our previous study that all animal cells have a membrane-bound nucleus. So remind yourself that the orange feature we have over there is our DNA, and that DNA is contained within a nuclear membrane. I'm not totally sure I've got the correct pink, but let's just go with it. A nuclear membrane. Okay, and we see that surrounding the DNA at the heart of this cell. Okay, now what I really want to be clear about with the nucleus, let me just be absolutely clear about with this nucleus, is that this nucleus controls the activities of the cell. I can't emphasize the importance of that enough. All of the cell's activities are controlled by the nucleus. So start to get to grips with that idea. Next, I'd like to introduce you to these little fellas. And these are what we refer to, let me choose a different blue here. These are what we refer to as mitochondria. And I referred to these briefly in the prokaryotic and eukaryotic tutorial, because one of the things we said is you don't find them in prokaryotes, but mitochondria are super important. Why? Because they are what we describe as the main site of aerobic respiration. Now we say main site because we've also got to include other processes of aerobic respiration, or certainly glycolysis, which actually occurs in other parts of the cell. But most aerobic energy is released through the release of energy in these mitochondria. Okay, so we've got mitochondria being the main site of aerobic respiration. Actually, one point, let me just point to these things. One thing I just sort of highlight to you here, that each of these has been sort of cut as a cross section. Can you distinguish that this one has been cut sort of flat across this mitochondria, and this one's been cut in the other plane, in the other direction? So it's actually something you might just want to get to grips with. You might actually see these two different profiles when you're labeling cells yourself. So I would encourage you to think about these. In fact, I'm also going to give you a bit of a measurement of mitochondrion. So mitochondrion is a singular. They tend to be, or it tends to be, in the region of 0.2 to 0.7 micrometers. Okay, so a really, really tiny structure, but by no means the smallest of our subcellular structures. And in fact, I did actually include, let me just drag over here, I did actually include a little illustration of a mitochondria over here. Okay, and you can kind of get a bit of a feel for it here. You see its sort of innards, or its internal mechanisms here, and we can see that it's kind of got folded membranes on the inside. So this is where the main process of aerobic respiration is going to take place, and they're depicted in this way on our diagram. Now, let's keep going. Let's also now introduce what are referred to as ribosomes. Now, ribosomes, in my rather terrible drawing here, are these little red dots. And little is an important word, okay, because these are absolutely tiny structures, and they are responsible for what we refer to as protein synthesis. Protein synthesis. So when you are labeling things like animal cell images, you may find these represented as tiny little dots, and that's what will allow you to know that they're ribosomes. Now, in future study, you might start to wonder and find out why they kind of seem to be connected to these branch-like structures. And I'll leave that for another day, but it's perhaps something that you want to be thinking about and looking out for when you study this in a bit more detail in the future. And finally, I just want to remind you that within this cell, we also have cytoplasm. Now, cytoplasm, I decided not to depict it here, but cytoplasm is all over the cell, so I could kind of like sketch it in here, but the cytoplasm is all over the cell. And our cytoplasm is what we might refer to as our liquid gel, or sometimes we might call it our cell fluid. And it's a really important structure because it suspends organelles. Our membrane-bound subcellular structures, such as our mitochondria, for example, are suspended in that cytoplasm. Okay, let's move things on. What I'd like to really, I suppose, both introduce and compare to now is the distinction between an animal cell and a plant cell. So let's move on to our plant cell. And I think we'll go through this a little bit quicker. So with our plant cell, a couple of key things I'd like you to know straight away. I want to introduce some distinctions for you straight away. Obviously, what we have here is our cellular membrane. But as straight away, we now have an outer layer beyond that cellular membrane. And this is what we refer to as the cell wall. Okay, so the cell wall being this structure here, and that cell wall contains cellulose. Okay, cellulose. And it's really important, the point I'm going to make here, really important point, this performs the job of strengthening the cell, or I'm going to say here, it strengthens the cell. Okay, so it strengthens the cell. Now, you might want to reflect on why plant cells need a stronger structure than perhaps some animal cells do, but that's something we'll look at another time. And next, we have this structure in here, and this is what we refer to as a permanent vacuole. A permanent vacuole. And again, it kind of looks like some kind of haze or puddle within the cell. But the point I'd really like you guys to kind of get to grips with this permanent vacuole, it does the following job. It keeps the cell, it keeps the cell, and I'm going to put this in inverted commas, turgid. Okay, turgid, one of the ways of describing turgid is congested or full. Okay, so it keeps the cell turgid. And again, it's kind of structural, so you might want to be thinking about that going forward as well. Let's introduce some further elements. Well, this looks familiar, right? We have some DNA genetic material, and we've got a nuclear-bound membrane around it. We have a nucleus. I think we're happy with that, right? So just to be sure, we have our nucleus here, and we have our genetic material, or what we're simply going to refer to as our DNA, within that nuclear membrane. Let's move on. We also have mitochondria, the site of aerobic respiration. We also have ribosomes. We looked at those in our animal cells as well, so that is consistent to our plant cells. And finally, we have something that's distinct to green plants, and that is a feature which we refer to as chloroplasts. Chloroplasts. Now, these chloroplasts, when I've got one here, let me just sort of scribble all the way across, and here. These chloroplasts, they contain a chemical known as chlorophyll, which is super important when we come to the process of photosynthesis. And the point I'm going to make at this stage is they absorb light for energy. They absorb light for energy. So we'll come back to the detail of that process in future tutorials. But for now, try to be able to distinguish between what we might recognize as an animal cell structure and a plant cell structure. Now, I've also given you a relative scale for each of these. Now, it doesn't really matter that we've got one on the X and one on the Y axis, or one horizontal, one vertical here. What I really want to get across here is that the animal cells, they tend to be smaller eukaryotic cells compared to the plant cells, which tend to be bigger in the main. Of course, we do have some overlap there as well. Finally, let's just make sure we're happy with our new vocabulary. Cell wall, and particularly cellulose, the outer lining of a plant cell, and that cell wall, as we've said, strengthens the cell. Mitochondria, They are the main site of aerobic respiration. Ribosomes, they are where proteins are synthesized. And I should really say, when we talk about synthesized, maybe I should have clarified that, it's where proteins are made. Okay, that's what we mean when something's synthesized. Why could I not say that properly? Chloroplasts, again, remind yourselves, contain chlorophyll and absorb light energy. Chlorophyll, a chemical within chloroplasts. Permanent vacuole, as we said before, a feature of plant cells that keep, or not all plant cells interestingly, but many, and they keep the cell turgid, or if you like, congested, full. Turgid, I've just mentioned. Organelle is a membrane-bound substructure such as a mitochondrion, for example. Thank you.